Donc, Laetitia, est-ce que tu veux qu'on aille de l'avant?
Oui, s'il vous plaît.
Parfait.
Merci beaucoup. Merci beaucoup, professeur Macandray, de nous rejoindre aujourd'hui. Professeur Rafik Naccache, on est très content de vous recevoir aujourd'hui sur le séminaire du CBS, le chapitre étudiant de la vie de Québec.
I will switch to English because we have many. It will be bilingual. Everyone is very welcome to ask questions in French or in English. We will conduct this this seminar will be presented in English, and it's very. I'm very glad to welcome Rafik Naccache. Rafik indeed obtained his PhD in 2012, and I remember that I was a young professor at the moment, and he and his supervisor from Concordia knocked to my door because they wanted to do some MRI with nanoparticles that contains lanthanides, and we did end up to inject animals in that project, and that was actually a really interesting project, very fun. We learned a lot, and I'm very glad to see that after a very successful postdoc at INRS in the group of Roberto Morandotti, he was hired as a tenure track faculty professor at Concordia. So he's at the Department of Chemistry and Biochemistry at Concordia University. He was recognized as a Petro-Canada Young Innovator and Concordia University Research Fellow. His newly established group, well, newly 5 years ago, you established your group 5 years ago, Rafik. Your program focused on the study of the fundamental properties of fluorescent carbon nanomaterials and hybrid nanosystems for the development of catalysis, sensing, imaging applications. I can also add that you previously, prior to your PhD, I think that you worked in a pharmaceutical company, if I remember well.
That is correct. Yeah, for 10 years at Merck Frost.
So you were at you worked for Merck Frost 10 years, and I think this is something really interesting that the students can know, because very frequently our students in the biomaterials chapter, they ask to talk and to contact to people who have been exposed to both sides of the research. So you first worked for 10 years in the pharma industry, and then you decided to do your PhD, and you decided to forward in research. So if you are if you are interested, or if you if students are interested to contact you, and if you are interested to not to mentor but to have conversation with these students, that would be this. You might receive a few emails or contacts by students, because we know that our students like to talk with people who are exposed to both sides of the research. And finally, I would like to add that you are a CQMF, Centre Québécois des Matériaux Fonctionnels. You participate in the CQMF network, and this provides opportunities for collaborations between the different Quebec institutions that participate in that network. And today you will present on the carbon dots, imaging, sensing, and beyond. So thank you for being here today.
So sorry, because now my puppy decided that he's going to cry. So I'm just going to put him on the couch. And this is the trials and tribulations of working from home and having a new pet, and somebody decides to leave the house. And all right, now we're back. So yes, indeed, I did work at Merck for 10 years. I did my degree actually, well, my master's while I was working at Merck. And then I even started my PhD while I was working there. And then eventually they shut down the center in Montreal, and they wanted me to move to the US. But then I realized that no, my calling is academia, and I devoted myself to academia. And I think it worked out okay at the very end. So, all right. So thank you very much for the invitation. So I thank the organizers from the CBS and the student chapter for having me come and talk to you. And I want to talk to you today about carbon dots and what we use them for. But before I do that, I want to actually go ahead and really talk about the the people that do the work that actually make things happen. So this is an outdated pre-COVID picture, because now with COVID we can't really go anywhere. We have a curfew, as you all know. So But so these are the people, some past, some present, that have really contributed. Collaborators who we really have established some very fruitful collaborations with, and which help us out in the science, and we mutually benefit from one another. And of course, funding agencies, because without funding agencies, we can't really do very much research or go anywhere to present our research, except now by Zoom, we can do it from the comfort of our own home. So we work with something called carbon dots. And for many of you who may not be familiar, you might ask yourself, well, what is a carbon dot? And we're talking about spherical carbon nanoparticles that are less than 10 nanometers in size. They're predominantly amorphous. So when we run the XRD, we usually see a just big amorphous halo. So they're a disordered form of a carbon nanoparticle. And when we look a little bit more into their carbon chemistry, we see that they're mostly sp2 hybridized. There's some sp3 character. And it turns out that we can actually change that by the synthesis that we use, and even some of the starting precursors that we also use. And if you're interested in knowing what they're made out of, what they comprise, it's mostly carbon, hydrogen, oxygen. And of course, you can start having nitrogen, you can put sulfur, you can put phosphorus, depending on what you include in your starting precursors. The nice thing about these carbon dots is that they're highly dispersible in water. Now, I've spent all my previous nanomaterial experience working with nanomaterials that were hydrophobic, that I had to work really hard to make them hydrophilic and dispersible in water. These have a plethora of chemical functional groups on the surface, and because of that, they are very highly dispersible in water. Now, and I asterisk this point, coming from a pharma background, I realize that words have meaning, and when we talk about cytotoxicity and biocompatibility of nanomaterials, I need you all to understand that when you read something, whether it's in my paper's presentation or in the literature, this is based on a limited subset of data. There has been no long-term studies, and every time you make a nanoparticle or a nanomaterial and you do some modification to it, for all intent and purpose, it is like considering it being something new. So based on the data we've generated, based on what's available in the lab, they seem to actually have very good cytotoxicity relative to the concentrations that we need to be working with. But why are they really interesting? I've only told you about them. Well, it's because we can actually tune their fluorescence. So we can go even from the UV all the way to the far red. And once we start looking into the far red, well, it becomes interesting because we can start thinking about doing maybe some imaging applications in biology. You can also get them to fluoresce in gels or in solid state. More recently, we were able to make them actually fluoresce completely in the solid state, which was something that was not very typical. And in fact, when my students showed me that, I was not very convinced. And I said, well, prove it to me, because I still don't believe it. So they took it, dispersed it in water. They wrote with a pen on a paper, and they let it dry. And in fact, I got an eerie looking message that says the Naccache lab, but at least to me, it proved that, they do fluoresce under UV light. But I mean, ideally, what we're trying to do is we're trying to go into that near infrared window. We're already here. We're knocking on the door. We're at about 650 to 690, but we're trying to push this further into the near infrared, because this is where there is a lot less absorption by water, by skin, by hemoglobin, and so on and so forth. And if we want to do some imaging, this could actually become very interesting. So we are really trying to push their fluorescence into that region. Now, how are carbon dots made? Well, if you were to today go to the literature and look it up, you will find some very interesting articles about you can make them from orange juice. You can make them from coffee grinds. So after you've had your morning coffee, you can recover the filter that has the coffee grinds, and you can make them. You can make them out of honey. You can make them out of lemon peel. Spoiler alert, you can also make them out of orange peel, out of wood products, and out of coconut shells. And I apologize, there seems to be a little animation issue. But you can also make them out of coconut shells. In fact, there is a publication in the literature, and I believe Journal of Green Chemistry, where they made it out of urine, and they cheekily they called it P dots. Now we sort of set a limit as to what we use in our lab, and we certainly do not want our lab to smell that bad. But all jokes aside, while all these are very interesting because they show that you can make these carbon dots out of virtually anything, the problem with working with many of these sources is that they're very heterogeneous. So a coffee bean contains about 180 different carbon-containing compounds. So if you were to try and make a carbon dot out of this, it is very hard to sort of come together to an understanding as to what is contributing to the formation of the carbon dot and how can we control its properties. So we're trying to make these properties more homogeneous and more controllable. So we like to work with things like citric acid. We like to work with things like sucrose. We can do it from amino acids, all more individual molecules that are well studied that we can actually start thinking more about how these carbon dots do come together and form. You know, and you can do this top down or bottom up. We like to work mostly with bottom up techniques where we start arranging things together to come towards the formation of that nanoparticle. So we use things like microwave chemistry, where you have very short reaction times. Basically, we're talking between 5 to 10 minutes, you have synthesized your carbon and nanomaterials. But we've also done a lot of work with hydrothermal reactors, where you know, allows the pressure to reach much higher points, which would be limited otherwise in a conventional synthesis or even in the microwave. But we also do some conventional synthesis, which doesn't require a lot of pressure. It really depends on the combination of the materials you start with, as well as the solvents and the temperatures that you're heating these solvents at. So a lot of times when we're doing reactions in the microwave, we're doing it in water, but yet we let it go up to 200 degrees Celsius. So we really see an increase in pressure associated with using, let's say, water at 100 degrees above its boiling point, right? So mostly thermal mediated. We typically work in the range between 180 to 220, sometimes even lower, 160 to 220. We actually tried making them even at room temperatures. And after a week, we managed to get some carbon dots, but they were terrible. We didn't like anything about them. And we sort of abandoned the idea of doing that for now, but we may revisit it in the near future. But one of the keys when you're working with carbon dots, and actually any nanomaterial in that regard, is that purification is a huge component, and it's one that is often overlooked in the literature. So I cannot tell you how dismayed I am when I'm reading an article and says, you know, it's basically that our reaction went to completion and no purification was required. Yeah, I've never seen that ever in any of my reactions, and I'm a pretty good material scientist, and I know how to make nanoparticles. Marc-André, maybe you know, we've made enough batches together. you know how I was, but yeah, there's a lot of work that goes into purifying your materials, and this again becomes a problem when you're really trying to understand the fundamental properties of your material. If you don't purify, if you don't clean up all unreacted materials or fluorophores or things like that, you wind up getting a very heterogeneous source. So we've spent quite a bit of time working on this, and now we've sort of figured out really what we need to do. And the thing is every time you change too much in your materials, you actually have to sort of retweak your purification, be it through centrifugation, filtration, dialysis, or combinations thereof. So with that said, what are really our research interests? We sort of have three main axes in my group, and I try and tailor that according to our students' interest. We have the people who are more interested in on the sensing side of things, so a lot of metal sensing, a lot of chiral sensing, and metal sensing could be interesting both for environment or for biological applications, because metals do have a role in biology, and quite often we need to understand or know how much metals are in a given biological environment, as this could dictate a certain outcome for the way that maybe cells behave or organs are functioning. So there's definitely room for it in on both sides. With chiral nanomaterials, as I'll show you, this was an idea that we had on a whim, and from there, now a new sort of a whole new line of research has developed in my group, because it turns out that we can start borrowing cues from nature and use that to make nanomaterials that have antimicrobial behavior. And you know, if not living, if you know, before the pandemic, this didn't convince you that this is important, the current pandemic maybe, you know, is something that could inspire more work for people to continue looking in antimicrobials. And if nanomaterials can play that role, that would be great. We also do a lot of work on optical and magnetic resonance imaging. I mean, these are quite fluorescent. We can have some of them after extensive purification. We can have quantum yields of up to 65%, which are really bright. And we're talking micrograms per ml. Marc-André, you'll probably recognize this image from our paper together. Definitely, we are also introducing paramagnetic ions on some of these nanomaterials, but we're also starting to look at, well, how can we stabilize the metals so that they don't leach out once they're introduced into a biological system. So we're looking at even potential for transmetallation and things like that, and how we can create a really stable you know, complex with that metal ion, instead of simply just showing that there's some MRI contrast enhancement and then moving on to the next thing. So we're really trying to build it from the ground up in order to design effective vehicles. And something that recently took over in my group, and it's basically biofuels. I won't talk to you about biofuels today, but I mean, essentially it's working on clean energy and how we can use carbon dots as extremely cheap catalysts for transesterification of oils to biofuels, and how we can even recover waste oils and we can convert them into clean biodiesel, which would meet in the interest for these fuels. So we start our sort of discussion with these dual fluorescent dots, and most of the carbon dots, like quantum dots, distant cousins, they usually have a single emission fluorescence band. And we were interested in, can we actually induce multiple fluorescence centers in our carbon dots? And there were initial reports that glutathione and formamide would lead to that, but it wasn't really clear in the literature. So we embarked on that journey trying to see if we can in fact do it. And you know, we make these materials, we look at them by TEM, and they're god awful to look at by TEM because they're amorphous. So when you put them under the TEM, they have terrible, you know, electrons are not diffracting. I mean, you have very poor contrast, and to be able to get any reasonable contrast, you have to work with a very low voltage on the TEM. So you typically do not get very good TEMs of carbon dots, but when we look at their properties, we see that they have multiple absorptions here, and you see some in the red. And in fact, if we excite them with 4 or 5 nanometer light, we'll see blue fluorescence and we'll see red fluorescence. We could also, if we want, simply just excite in this region here, and we see red fluorescence. So we were very excited about this because you can get blue fluorescence, red fluorescence. So now this is starting to, you know, look different from what we've previously seen for carbon dots. And of course, my student shows me that, and I says, yeah, but, you know, please go and look at, you know, your starting materials. Make sure that there is no absorbance, that this is not basically something that's left over from our reaction. They go through great lengths to prove everything to me. I am satisfied by this. And I'm a material scientist, and I want to know, you know, how can I really tailor this fluorescence? Because once we start tailoring the fluorescence, we can start thinking about how we can, you know, apply this. And it turns out that by simply manipulating reaction temperature, you can actually tailor this red to blue fluorescence very much. So you can go from predominantly red to, you know, somewhere 1-to-1, and just completely blue, and you would kill your red fluorescence. So really, I guess, depends on what you'd want to do. And if you fill in the blanks, you can get a whole spectrum of these colors. But of course, at this point, we're trying to understand, well, that's great, but how are these forming? Again, I need to understand, and this is something that's very important, you need to understand, you know, more about your material before you jump to the applications. So I'm going to show you applications, but those come after, you know, a lot of groundwork that builds up our knowledge and understanding of these materials. So typically, a carbon dot reaction mechanism undergoes a series of, different steps where you start off with precursors that then fluorophores, then you have a polymer, then a polymerization, and then you form a carbonization where you get these carbon dots. And the reason why we think this happens is because we've relied on chemistry, and we're not the only ones who are obviously proposing this. This has been also proposed by other researchers working in carbon dots. Is that some of the studies, like for example, when we start off with a citric acid molecule and an amine precursor, some of these, like ethylene diamine, for example, some of these have been known as reactions in the literature. And in fact, the formation of intermediates have been isolated, looked at by mass spectroscopy and by NMR in some cases. So we've done a lot of work with NMR and mass spec as well in order to be able to study some of the intermediates that form. So we know that this is what usually happens. You wind up forming these heteroaromatic intermediates, which then would come together. And then they would undergo a carbonization, polymerization, and then or cross-linking, depending on the reactions that you are looking at, and eventually you form this carbonized material. But typically, we only see blue fluorescence out of this aromatic carbon dot, and in fact, I would tell you that 98% of all publications you'll find on carbon dots, they report blue fluorescence. So how do we get the blue and red fluorescence? Well, so we were very much interested in that. And, we thought, okay, well, if we're reacting glutathione with formamide, we're probably forming some intermediate fluorophore, which is then going to start following it's going to be followed by the formation of a carbon core, which is very typical to what you see for carbon dots and probably results in the blue fluorescence component. Now, there is going to be growth of the core, and as the core keeps growing, you are going to be consuming these fluorophores. And what's likely happening is that we have two different states. We have the core state, which is resulting in the blue, as well as the molecular states, which have this red fluorescence that is occurring at the surface. And this was a very interesting idea. We have now also we have submitted. So this is out of date, this reference, but we wind up showing this by basically ultra-fast spectroscopies at the femtosecond scale, that in fact there are two different states coming, and that the molecular states are responsible for the red fluorescence. But at the time when this happened, we wanted to think of a very quick way of proving this to ourselves. We said, okay, well, if one is the core state and one is the surface or molecular states, we would expect them to respond differently to their environments, right? They would have different sensitivities because typically the core would be more sheltered by the surface. So why not test if they are sensitive to things like temperature, pH, heavy metals? Let's see what happens when we change their environments. If in fact it is a core versus a molecular or surface state, we should see differences in the way that they respond to their environment. And we said, okay, well, the easiest thing we can do, because we have a fluorimeter and a Peltier controller, we can change the temperature and run it at different temperatures and see what happens. And, if we want to do that, we're trying to do temperature sensing at the nanoscale. It's been used in the literature. The term is known as nanothermometry. And you're using nano-sized fluorescent materials. They're highly interesting for things like biological imaging and diagnostics. But what they do require you to do is have some kind of change in the fluorescence signal, right? So these are all different ways that you can do it. It could be changing your spectral position. It could be changing your band shapes, right? It could be changing your bandwidth. Things go broader or narrower. It could be changes in intensity. It could be changes when you change the polarization of the light or lifetime measurements. So basically we asked ourselves, well, can our dual fluorescent dots be used for temperature sensing? But first, let's find out if, this blue and red are actually changing as a function of temperature. And sort of a spoiler alert of what's to come, we look at two ways that we can do this: changes in the band shapes as well as changes in the intensity. And I'm going to talk to you about why one of these two is not a good way of doing any sort of measurements. So Jinray, who's my PhD student, went on in the lab, did these experiments for a while, and what he sees is that, in fact, as you increase the temperature between 5 and 60 degrees Celsius, you see a continuous increase, excuse me, in the red fluorescence, and almost, if you will agree with me, no decrease in the blue fluorescence. Also, if he decides to excite just in this region here, where you only see the red fluorescence, he sees the intensity increases. So already this is telling us that something is happening. He comes back, he shows me these beautiful linear curves, and I say, this is fantastic. I am very happy. However, you should repeat it again. So he leaves my office, he goes, he repeats it again, comes back and shows me the same thing. And I said, well, I like the fact that you're showing me very linear stuff, but it really bothers me because I don't see any error bars. You should go repeat it again. He leaves my office quite upset with me, goes, he repeats it, comes back and shows me the same thing. And then I told him, well, you have to show me where the error bars are. And it turns out, in fact, that we do have error bars on that, but they're actually quite small that you don't see them. So they were actually quite reproducible in terms of the way that they behave to temperature changes. Now, the nice thing about that is that when we change batches, when we make a new batch of nanomaterial, the response is there, and it's not going to behave very differently, right? Unless you've changed the way that you make your nanomaterials. And in fact, the thermal sensitivity is almost it's 0.05 kelvins to the -1. So that means that, basically we're looking at very small event changes, which we can register using fluorescence. And we were interested in seeing how long we can actually store these nanomaterials. And for a period of 6 months, we don't see any significant fluctuations. And when we do cycling of heating, cooling over several cycles, we did more than that, but I think I'm only showing 6 or 7. We don't see any change. We see the fluorescence recovering back and forth. And we're not talking about leaving it for hours here to recover. It recovers almost instantaneously under a minute, essentially giving time for the solution to equilibrate back and forth. So we thought, well, this is really cool. But my sort of objection to showing that this works in the cuvette is that everything works in a cuvette. It's very controlled as an environment. And a lot of times we, in material science, when we want to propose an application, we need to go above and beyond the cuvette model. We need to really show that this is something that can work. So in that sense, I told him, this is great, but let's start looking at if we can actually do this in a controlled biological system. How about we do it in cells? And we picked to work with HeLa cells. We had a collaborator upstairs in the biology department, which was willing to collaborate with us on that. And we got trained in how we can maintain these cell cultures and how we can incubate. And we wanted to do some temperature sensing. But of course, safety first. We need to know what happens when we put our carbon dots with HeLa cells, right? Are we simply going to add the carbon dot and the HeLa cell is going to die? Because if that's the case, there's no point doing the experiment. So we did a lot of safety studies. And we determined, that our IC50 was about 150 micrograms per ml. We are working at 25 micrograms per ml, or even lower. So it's significantly below our IC50. We're not really coming close to the point where we're going to be killing the cells on our own. So we incubate our carbon dots in there. We see that they have been taken up. We've washed, of course, to make sure that whatever carbon dots are still floating around in any medium or anything like that have been, taken care of. And what we see is that, we allow them to incubate at different temperatures. And then what we did was we measured the blue and red response from the fluorescence of the nanoparticles using filters. And we did this over hundreds and hundreds of measurements. Of course, I can't show you hundreds and hundreds on the same slide, but essentially to show you that wherever we'd measure blue, we would also have the filters and we'd measure the red fluorescence. And we looked at the ratio of the red to blue fluorescence. And once again, we see that as you increase the temperature, you see that the red to blue fluorescence increases, which is similar to what we saw in the cuvette. So we were actually quite happy about this because the same trend followed. Remember that we could also do this by intensity. So in this case, we're doing it ratios, right? We're taking a ratio of the red to blue. If we do it by intensity, it doesn't work because cells don't behave very well at 42 degrees Celsius, and they don't behave very well at 32 degrees Celsius. So the intensities that we were measuring were simply in response to the cell and how it felt about uptaking a carbon dot at 32 degrees or at 42 degrees. So the measurements were actually could not be correlated to what we saw. But by having a ratiometric way, you have an internal self-reference because our blue fluorescence did not change, right? So basically, whatever was happening in terms of the changes, we were always correlating it to the blue fluorescence. And because of that, we actually observed something that's quite similar relative to, our cuvette model. And when we image the HeLa cells, and what you see here in red is our carbon dots. And they're located somewhere near the Golgi apparatus, which, I mean, we expected that that's where they go. They're basically, getting compartmentalized in this location here. Now, what this has led us to sort of move on with our studies is also trying to understand if we can track these carbon dots and then where they go in the cell afterwards, and then what is their fate after this. So can we use them to actually track events in the cell? So we're currently collaborating with our colleague in biology on that with a graduate student. So we said, well, this is great. We can do temperature, but is this a one-trick pony type thing, or can we do something else? Can we maybe measure changes in pH? So we said, okay, and this is work that was done with Adrian, who's now doing the joint masters with biology and myself. Essentially, let's look at the changes in pH, and then we look at changes in the red to blue fluorescence. And, we sort of didn't see very much. I mean, the blue was not changing. The red was kind of changing, but boy, I would have a hard time standing here and trying to defend very small changes like this, telling you that I think, we have sensitivity to pH. We did it by quantum yields. We looked at the fluorescence. We even looked at lifetimes. We're not really seeing very much. So we said, okay, well, maybe they're not really sensitive to pH. But we did notice something. We saw that there are changes in the shape of the red fluorescence band. So the red fluorescence band normally has a shoulder, and then as you change the pH of your medium, we started seeing amplification of that shoulder, and where basically the intensity of that shoulder would increase at the expense of the peak. So we have a shoulder here at 650 nanometers, which would grow in intensity, while the band at 680 would decrease in intensity. So once again, we were able to see that we can do this ratiometrically by measuring the changes in the fluorescence intensity bands at 650 to 680. One is decreasing, one is increasing, so they're self-referencing relative to each other. And in fact, we do have sensitivity to changes in pH between about 4 and 7, which, I mean, is not a huge range, but it turns out that this is actually good enough for what we wanted in terms of a biological sort of to study changes in biological pH medium. And at the same time, Dusika Masinger from McGill comes to give a talk. We meet and we discuss that she was looking to use a nanomaterial to measure the change in pH inside glioblastoma cells following the administration of combination therapy of cancer drugs. He said, okay, so let's try it out. So it turns out that in cells, we can probe this. She was interested in administering things like bafillomycin, metformin, and diclofenac because there were reports in that case of the increasing lysosomal pH in cells when you do this, and we're looking at the change at 680 to 650. Yeah, I think the reports were for diclofenac that there's an anyways. So we were trying to really measure that change in pH inside the live glioblastoma cells. And in fact, we incubated those in the glioblastoma. And once again, we measure the fluorescence intensity. This one was a bit more difficult because we were trying to actually filter out the 650 versus 680. So it was a little bit challenging, but I have to say, Isan, who was working with Lucica Maysinger, and Junrei, who was working in my group, worked very closely together on that. And what they actually were able to show is that here's your control and here is the ratio. And when you administer bafillomycin or metformin, you actually see that there is going to be an increase in the pH, because you see the ratio at 680 to 650 is in fact decreasing relative to what we would normally expect based on that sigmoidal trend that we saw. Whereas with diclofenac, it does actually it doesn't actually change that much. And of course, when we compare that to starvation, which is always used as a positive control for lysosomal acidification, we see that it increases because the ratio of 680 to 650 is very low. So therefore, it will not obviously, it will not be as low. So we were able to show that bafillomycin and metformin can be used as potentially interesting combination molecules for glioblastoma. And we can use that to modify the pH inside a cancer cell. And if you do that, this is potentially a good and effective way where you can actually disrupt how the cell is working. And from there, we were also interested in in how these carbon dots can potentially interact with their environment if we have any metals present. And this is work that was done by Francisco, who is now who went on to do a master's and then now is doing a PhD in Toronto, and Adrian as well with Junrei. And what we see is that when we introduce metallic cations into a solution, we see a quenching mechanism that is happening where the red fluorescence is quenching and nothing is happening to the blue. This is still supporting everything we've come to so far about how the red is likely coming, the red fluorescence is coming from these surface states, whereas the blue is more from the core that is being shielded by the surface of the carbon dots. And in fact, if you were to look at the change in red to blue ratio as a function of concentration from half a nanomolar all the way to 1,000 nanomolars, it is pretty darn linear. And you could also do this with absorbance, because with absorbance it's really cool. You actually see the appearance of a new band that is associated with the complex of the formation of the metal cation and the carbon dot. So in one case we see the appearance of this complex versus the quenching that results in the decrease here that we observe. And our LODs are about 37 nanometers. We also do this for mercury, and we observe something very similar where, it's about 39.5 nanomolar, excuse me, I said nanomolar, and it's quite linear. And fluorescence works really well to quantitate metal cations. But the problem is, whether I show you this or show you the one on the previous slide, you can't tell what metallic cation is present. It could be lead, it could be mercury. Also, what happens when you have multiple ions present? Because it turns out that in real life, we don't work in very controlled environments, right? There is multiple metallic cations. And by the way, we're just working with, as a proof of concept, we're working with metallic salts. But in real life, metals don't exist as metallic salts just floating around. They're complex to dissolved organic matter. So, but you have to build it up. You can't learn to, race a car if you don't even know how to build an engine and then design the car. So, it's all about building it up. So, we also wanted more insight into the quenching mechanism. So it turns out that if you use fluorescence, it's great, you will get your answer, but you don't know what metallic cation is present. But if you use absorbance, you wind up getting a different response depending on the type of metal that you have. So metallic cation for mercury, you see this appearance of a new band here. As this decreases, this increases. For lead, you see basically a completely different response where this will completely quench and this will now appear in response to the formation of a new complex between lead and the carbon dots. And once you start mixing them together, you start actually seeing the different bands that appear due to the formation of a complex with lead and mercury, but they start becoming very hard to quantitate. So it becomes more a qualitative tool more than a quantitative tool. Excuse me. But one thing that was very interesting is that we saw selectivity for lead and we saw selectivity for mercury. And believe you me, we tried many different metallic cations. We actually tried more than what I'm showing you here, but just for the sake of clarity, I'm showing you a fraction of them. We see significant quenching with lead. We see significant quenching with mercury. We've gone back and we've done some LODs. We've seen some 22 and 28 parts per billion, which is similar. If you can report in nanomolars or part per billion. I know previously I reported in nanomolar. Both have shown static quenching based on the appearance of that band that we see, based on temperature studies that we have done, where as you increase the temperature, you see that it actually breaks apart the complex of the metal and the carbon dot, based on the fact that we've measured the lifetimes and we see no changes in the lifetime whatsoever. So we know this is a static quenching mechanism. And surprisingly, while they're not difficult studies to do, often there's a lot of hand waving that goes on in the literature about some electron transfer happening between the nanoparticle and the metal cation, but we're not really seeing much of that. Okay. So, of course, that begs the question, well, why are we only observing, selectivity with these two metals? Or why is it so sensitive that they quench our carbon dots, but we don't really see this with any of the other ones? Well, we went back and we looked at the surface characterization that we have done of our carbon dots, the ones that were formed from glutathione and formamide, which we have used for these studies because these are the dual fluorescent ones. And we know we have thiols on the surface of our carbon dots. We have thiophene groups, and we have a plethora of other functional groups. But one thing we know is that there's a certain affinity to sulfur by lead and mercury. We know that they interact strongly with sulfur-containing compounds. And we actually went to something really simple, like the hard and soft acid-base theory, which we teach in inorganic chem in our first year. And we said, okay, well, usually when you have a hard versus a soft acid, a hard versus a soft acid or base, you have things like mercury and lead, which are considered as soft acids. And these will bind to soft bases. They'll bind preferentially to strong bases. And it turns out that thiol groups tend to be soft bases. So the soft acids are going to bind to soft bases. It makes perfect sense, that they would do that. But then we looked at some of the metals we tested, and we said, well, some of the other metals also fall in that borderline soft acids, but yet they didn't quench very much. So it seems that there is also something that has to do with charge density. So when we looked at some of the metal ions, the cations that we looked at their charge, and we looked at their ionic radius, and we looked, at the charge density. And what we actually see is that lead and mercury have the lowest charge densities among a lot of the other metal cations. So it is likely, because of this low charge density, that they did not experience repulsion, let's say, as they approach the carbon dot surface, right, which allows them to interact mostly with the thiol groups. So now what we're going to look at is by protonating or deprotonating the surface of our carbon dots, we'd like to be able to compare how some of the other metals with the higher charge densities will do. And then based on that, we can, maybe that will inspire additional applications as to, how we can design better metal sensors, particularly if we're interested in using them either for biological or environmental sort of applications. Now, the last segment, and I know I may be going a little bit over time, so please do stop me if I am. I wanted to talk to you about, and I'll try and go relatively fast with this, is about chirality. So we were very interested in making chiral carbon dots, and many of you probably know about chirality, so I'm not going to necessarily go into great detail, but essentially, if it's chiral, I cannot superimpose it. And nature is chiral, like it or not, right? We see it in biological systems, in the twists of, DNA, in amino acids. We have L amino acids, D amino acids, sugars, D ribose, L ribose, in actually receptor sites on proteins, right? So they recognize chiral molecules, and that's why R-limonene smells like oranges and S-limonene smells like turpentine, okay? So because of these different chiral molecules and how they interact with receptor sites in our body. We know chirality is really important in pharma. If you don't believe me, the classic example often given is thalidomide, which was given to pregnant women in the 60s. One was sleep-inducing, one resulted in teratogenic birth defects in children. But we also know that in some cases in pharma, there are racemic mixtures. So for example, ibuprofen is racemic. So when you take ibuprofen, it's both the plus minus racemic mixture. So what we're interested in is, well, nature responds differently to chirality. Can we design chiral nanomaterials? And I know chiral nanomaterials have been done, and I know that you can do it post-synthesis. You can functionalize whatever chiral material you want. But we were interested in making it chiral to begin with. And if chirality is at the center of what we're interested in, we have precursors, temperature, time, molar ratios, pressures, all of these things that come into play. So what we're interested in is, well, how can we design a material like that? So we said, okay, we're going to start off with citric acid. Citric acid is achiral, but we're going to add a chiral precursor. So what could maybe be something that we can easily add, and hopefully we form a chiral carbon dot? Well, we're going to pick an amino acid. So we looked in our cabinet, we had cysteine, we had plenty of L-cysteine. L-cysteine is really cheap. We had some D-cysteine, it's really expensive. It's about a few hundred times more expensive. So we said we're going to do all our work with L-cysteine, and then once we figure it out, we're going to make D-cysteine, okay? And we're going to use water. That's the only solvent we're going to use. Water is not going to contribute any carbon. It's achiral, so we're perfectly happy using that. And we go ahead and we do the synthesis. Our particles are between 8 to 16 nanometers majoritarily. They look very different from our starting material. We look at the XPS as well to sort of, see what exactly we have on the surface of our carbon dots, because we don't know what's going to come later. But we, in fact, we also, after we did all of that, we also did the D-cysteine, and we don't see any differences when we make an L-cysteine carbon dot or D-cysteine carbon dot with respect to the functional groups, with respect to the size, with respect to the XPS. So for all intent and purpose, they look the same by physical characterization techniques. We know that, we're always interested in fluorescence, and we sort of had to look at the effect of the synthesis temperature on the fluorescence. So we don't see any changes in the fluorescence, but we do changes how you can change your fluorescence intensity. And it's going to turn out that, I'm going to tell you from now that when we were doing this, we sort of had to make a trade-off between how much fluorescence intensity we have, basically quantum yield of fluorescence, versus chirality, because you basically raise the temperature too much, you're going to kill the chirality in your carbon dots. And whether, again, you're doing it with L or D-cysteine, it's exactly the same. So is it chiral? Yes, actually, it turns out to be chiral. And we see a completely different response relative to your L and D cysteine precursors. So this is, you know, if you do basically any circular dichroism measurements, this is the response from your nanoparticle. This is response of your starting material. And the beauty about it is that the response from the L and D are actually opposite to each other. So we were very excited about this. And if you play around with the reaction conditions, you can actually tune your chiral response. You go from 1 to 1. If you have more citric acid, so less of the chiral component, you will decrease the chiral response, or it can go the other way. Same thing with D. You can have 1 to 1, 1 to 2, or 2 to 1. So if I increase my chiral precursor, I'm going to have a greater chiral response. So one of the things we're interested in, because we knew that, you know, we're borrowing this from nature, right? The whole L and D idea. You know, we had come across reports that E. coli tends to actually not do too well with when you incubate it with D-cysteine. So we said, well, we have D-cysteine carbon dots and we have L-cysteine carbon dots. Well, can we do something with them? And in fact, it turns out that, you know, when you get to concentrations of 1 milligram per ml, you can actually inhibit the growth of E. coli. Now, we've also tried this with other bacterial lines, and in some cases, we're even able to go to lower concentrations of carbon dots. So it really depends on the response. Now, this is really the first time we've done this, and we're working a lot more on it now. Again, we're trying to understand the mechanism as to why this is occurring, because once you understand what is really happening, you can design a much better chiral carbon dot that will have antibacterial, antimicrobial activities, and you might actually require a lot less concentrations than, you know, some of this preliminary work that you're showing. But now this is opening up a whole sort of ideas, new set of ideas as to we can probably, you know, think about smart packaging. We can start thinking about the antibacterial implants. You can start thinking about antimicrobial surfaces. You know, more so than ever, I've used Lysol so much in recent times that maybe if we have an antimicrobial surface, maybe we can, you know, use a little bit less of it. You know, if it's basically self-cleaning. So with that, I will stop my talk. I hope I convince you that carbon dots are versatile optical probes, that their optical properties can be quite attractive for sensing applications. We talked about temperature, pH, and metal sensing applications. That dual fluorescent carbon dots that have a ratiometric, we can use sometimes ratiometric means, and that basically allows us to introduce a self-referencing measure, that they can be developed as multifunctional sensors. We're trying right now to simultaneously build a pH and temperature sensor at the same time. And by taking cues from nature, we can design chiral nanomaterials that have these antimicrobial activities. So I will stop here, and thank you for the invitation, and thank you for your time and for listening to me. I'm sorry I went over by maybe 10 minutes.
No, there's no problem. Thank you very much, Dr. Naccache, Rafik, for this great and inspiring talk. Very, very good. I enjoyed it very much. I would like to open the session of questions, and so first to students and other participants. You can use the wave hand button so that I'm sure that I can see you. So I can start with one first question. I had have you all of these functions, the carbon dots have different functions, and probably one of the next step will be to integrate that function into an encapsulation or an embedding that allows the function to be part of an object. Could you comment on the difficulty to bring these the difficulty or specific challenges to bringing these carbon dots into films or fabrics or hydrogels? Are you facing certain challenge when it comes time to attach the particles to solidly and fixed into an object?
Yeah. So we thank you for your question. That's a really good question, actually. So we've done it where we've incorporated them into polymers. right? One of the challenges was making sure that, for example, at the time we were interested in looking at potential drug release, right? So if the carbon dot could be loaded onto a polymer vehicle, which then could be triggered to release the carbon dot carrying a therapeutic agent. And yeah, one of the challenges was the release, but with clever chemistry and sometimes not very complicated chemistry, we were able to get actually the carbon dots to release quite well from the polymers, right? But one of the things we do notice is that we might wind up impacting, because if we're talking about surface states or core states and things like that, we might wind up sometimes affecting their optical properties when we do that. So it really depends on the situation, but sometimes it's a bit of a trade-off. You might be able to, but you might also wind up impacting some of the properties. Now, the nice thing about being able to incorporate them or integrate them into like a device or a vehicle or something like that, is their surface chemistry of the carbon dots is so versatile that it allows us to do all of that. So in that sense, we have that flexibility, but we just have to make sure that we don't wind up sort of silencing some of the properties that we're very interested in.
Yeah, I had in mind a specific application where, so if, for instance, you would like to put the fluorescent carbon dots for temperature and pH sensing into a petri dish. So of course you can put the nanoparticles inside the cells, but what if you put them just under the cells and you use the color for thermometric purposes, or of course you always have the possibility to put a pH meter or to test to dip a paper on that, but there might be potential applications where looking at the petri dish in order to see if it changed color, direct indicator of the temperature of the very surface on which the cells are sitting. I don't know if it would be possible. Or what's
if they're in solution, we're okay. We can do it as long as they are dispersed in whatever the solution is, or the growth medium. Because most of the work that we've done actually this was in the cell growth medium. So it's not like we did it in water and then now in the cell growth medium. No, this is directly. They remain stable. They don't precipitate. So we would be able to do it. In the solid state, it might be a little bit more difficult. So as long as we remain in dispersion, we can actually probably measure it relatively well.
Do we have questions from the audience? Yes, Henry, and then Samira.
Thank you very much, and wonderful presentation that you provided.
Thank you.
I've learned so much from carbon dots and its various applications. I was really interested in the chirality of carbon dots with respect to antibacterial and antimicrobial applications. Yes, obviously this is a new line of research, and so the mechanism may not be very, like you said, you're still working on it. Yep. And from the results you gave us, with it, I'm guessing it's from gram-negative bacteria, E. coli.
Yeah.
Did you perform any tests for gram-positives? And how are the results?
Yeah, in our paper we actually did, and we saw in some cases as well some response to that. However, like I said, unfortunately, COVID came so this paper was sent off for publication near the beginning of the pandemic, and then the student wound up graduating during the pandemic. So unfortunately, we didn't get the chance to, we're gearing up now to be able to look at some of these findings. I mean, we had a couple of theories, but honestly, nothing that I would, be very firm about right now. I think they would be very speculative at best. Yeah.
Thank you very much. That's interesting presentation.
And Samira?
Hi, Professor Naccache. Thank you very much. Very interesting presentation. I was wondering about the application of carbon nanodots for biodegradable implants to detect the ion release. I saw that you showed a graph related to, for example, nickel, cobalt, positive ions that you were able to detect with carbon nanodots, but they were in a similar range, right? I was wondering if we, for example, if we have both the cations in the solution, how we can detect them first, and if carbon nanodots can be used for biodegradable metals. Thank you very much.
So thank you for your question. So basically, you're very right. You saw this, right? But remember that, you know, based on what we have come to now understand about this, is that it really depends. Charge density seems to be a very important factor because it's going to affect the interactions of the different functional groups you have on your surface. So we have carboxylics, we have amines, we have thiols. I mean, and you know, this is sort of what we're investigating now, right? By changing the pH of our medium and by protonating versus deprotonating certain functional groups, what we are expecting to see is that we will start showing the possibility of being able to differentiate between different, you know, we're going to see the impact, the quenching effect of these different metal cations on the carbon dots. Now, how do we know that that will happen? We know that by absorbance spectroscopy, right, we start seeing a different signal response based on the type of complex between the metal ion and the carbon dots, and they don't all show the same absorption spectrum. So for example, you see the position here for lead versus the position here for mercury. We can actually look at these differences and we can identify which metal cation is present. So in fact, when we mix mercury and lead together, while I mean it's not a huge change that you see, you see this double peak here. This actually corresponds to mercury and corresponds to lead. So we can start using it more as a qualitative tool. We can try and quantitate by deconvoluting, but when the signal response is very weak, it's really hard for me to sort of promise you that we'll be able to do that. We also have to be realistic. But I mean, if we can have if we can have a tool that can even tell us about the kind of metal cation species that are present in solution, this is great. And if we can do this in a biological medium, this is even fantastic, right? Because it's not very difficult to actually do these measurements.
Exactly. Thank you very much.
No problem.
And I think this will conclude the seminar because we are now over the hour. So thank you very much, Dr. Naccache, once again for this great talk.
Thank you very much for the invitation and for the time.
And thank you to CBS for the invitation. Thank you, Samira. Thank you, Laetitia. And if the students have questions, you can send your questions to me or to Laetitia or to Samira, and we will channel the questions to Rafik for if you want to continue conversations, or if you have specific questions on the talk, it will be a pleasure to do so.
Yeah. And if ever later on you want to reach out, please, you have my email, go for it.
Donc, merci beaucoup. Donc, ça va conclure pour aujourd'hui la session de présentation et le séminaire de CBS aujourd'hui. Merci beaucoup à tout le monde d'avoir participé.
Merci beaucoup. Merci à vous.
Thank you.
Merci. Thank you.
Ouais.